appear mucoid. Appear mucoid. They have mucoid appearance because of the production of polysaccharide capsule. These strains are particularly common in patients with cystic fibrosis. Some species produce or form diffusible pigments. For example, pyocyanin, its color is blue. Pyoverdine, its color is yellow-green. And pyruvine, its color is reddish to brown. That give them a characteristic appearance in color and simplify the preliminary identification. It helps scientific to recognize or identify the bacteria. For example, we have some bacteria in laboratory, but Pseudomonas produce some pigments in the culture media, and it helps the laboratory man or woman to identify it. Of course, it is very useful. And the other point is Pseudomonas aeruginosa has one of the largest bacterial genomes, encoding about 5,567 genes, including 568 regulatory genes. This is important for the understanding that Pseudomonas aeruginosa is highly adaptable. It can adapt itself to any environment and is capable of growth in a wide range of environmental conditions and in the presence of antimicrobial. You can see here it is very useful for this bacteria. You can see this picture here. It's a picture of Pseudomonas. You can see polar flagella, one flagella at one side. You can see pili here. Sorry, lots of pili around it. Pili helps bacteria to attach host cells. You can see LPS or lipopolysaccharide. As you know, Pseudomonas aeruginosa is a gram-negative bacteria, and of course, definitely, it has LPS in its cell wall structure, and it produces segregated some exotoxins, some enzymes. You can see here some enzymes, protease, phospholipase, some siderophores for capturing or binding to iron. And we can consider these factors as virulence factor. You can see here, Pseudomonas aeruginosa has many virulence factors, including adhesins, chili, flagella, LPS, are adhesives, toxins, enzymes, type 3 secretion system that we will speak about it with details in next slide. Now we want to speak about adhesives. Of course, adhesives are really important because after a bacteria, a pathogen bacteria is entered to your body, in first step, it should be attached to host cell. As with many bacteria, adherence or attachment to host cells is critical or important for establishing infection. At least 4 surface components of Pseudomonas aeruginosa facilitate or help this attachment. 1, flagella, 2, pili, 3, lipopolysaccharide, And 4, alginate. Flagella and pili, in addition to attachment, help or mediate motility in Pseudomonas aeruginosa. It means they have 2 responsibilities. One of them is attachment, and the other one is motility. Lipid A is one of the components of LPS and is responsible for endotoxin activity. Endotoxin activity is a virulence factor of Pseudomonas aeruginosa. And alginate, the first one, is alginate. What is alginate? Alginate is a mucoid exopolysaccharide that forms a prominent capsule that makes a prominent capsule on the bacterial surface and protects the organism from phagocytosis and antibiotic killing. Production of these mucoid polysaccharides is under complex regulation. The genes controlling production of the alginate polysaccharide can be activated in patients such as those with CF means cystic fibrosis or other chronic respiratory disease who are predisposed to long-term colonization with these mucoid strains of Pseudomonas aeruginosa. So alginate is very important. Alginate is an exopolysaccharide which covered the surface of the bacteria like capsule. You can see here in this slide. Please pay attention to this slide. This is the membrane and cell wall of the bacteria. This is outer membrane of the bacteria, and this part is an inner membrane of the bacteria, and this space is preplastic space. This space is preplastic space, and this part is surface of the bacteria, and alginate is placed on the surface of the bacteria. Alginate is exopolysaccharide, mucoid exopolysaccharide. Alginate is mucoid exopolysaccharide or has mucoid appearance, is made of exopolysaccharide and covered on the surface of the bacteria. And as I told, we have some genes like C, A, D. We have some genes in the bacteria for regulating for expression of alginate in the bacteria, and the expression of alginate and the production of alginate is affected by some factors, some hosts, cell, or some bacterial cell. So as I want to summarize these points, we have some genes for making alginate in the bacteria. They are responsible for producing alginate and for making alginate on the surface of the bacteria. Alginate is composed of exopolysaccharide, and they cover on the surface of the bacteria, and it has mucoid appearance. It helps the bacteria to attach to the host cell. Does this? No. Besides, or in addition to attachments, alginate protect the bacteria from phagocytosis, from macrophages, from antibiotic killing. How? I will show you in next slide. Look at this picture. You can pay attention in the left side. This bacilli, the road shape is bacteria, and this one is a phagocyte cell, like macrophage, like neutrophil, like natural killer cell. A phagocyte cell. Phagocyte cell like macrophage. This macrophage is a part of our immune cell, can kill this bacteria, can phagocyte this bacteria and do phagocytosis. You can see here, do phagocytosis. But you can see the right side, around this bacteria, you can see a green circle. You can see a green circle. This green circle indicates a capsule. This green circle indicates a capsule, indicates an alginate, something like this, indicates an exopolysaccharide. So, it protects bacteria from phagocytosis. The macrophage cannot recognize bacteria due to capsule. This macrophage doesn't recognize the bacteria and cannot kill the bacteria, cannot phagocyte the bacteria due to its capsule. So the capsule, the alginate, the exopolysaccharide protect the bacteria and help the bacteria to be alive, to be survive. Salam. Welcome to the class. So, alginate have two responsibilities. One of them help the bacteria to attach to the host cell. The other one protects or helps the bacteria against phagocytosis, against immune response of host. Do you understand? Do you have any question about it, about the adhesins? So, as I told, we have, okay, that's very good. We have 4 adhesins: flagella, pili, alginate, and LPS. Okay, that's very good. Okay, now we want to speak about exotoxins. In Pseudomonas species, 3 types of exotoxins are recognized, are identified. One of them, which are the most important, is which is the most important, sorry. Exotoxin A or ETA. ETA is believed to be one of the most important virulence factors. Not just the most important exotoxin, is the most important virulence factors produced by pathogenic strains of Pseudomonas aeruginosa. How? What is the mechanism action of this toxin? This toxin disrupts protein synthesis, block protein synthesis, stops protein synthesis. How? By blocking peptide chain elongation in eukaryotic cells, much like the diphtheria toxin product produced by Corynebacterium diphtheria. However, the toxins produced by these two organisms are structurally and immunologically different, and ETA is less potent than diphtheria toxin. Exotoxin A is similar to diphtheria toxin, although their structure are not similar and are different to each other, but the mechanism of their mechanism are similar to each other. Both of them disrupt protein synthesis in eukaryotic cell. And as I didn't find any good picture related to the Pseudomonas aeruginosa, I put this picture here. Please pay attention to this picture. In left side, you can see normal protein synthesis. In left side, you can see normal protein synthesis. In eukaryotic cell, this is ribosome. And in the right side, you can see protein synthesis stops. Protein synthesis stops. In left side, in the ribosome, amino acids are attaching to each other one by one. Amino acids are binding to each other one by one through or by using EF2 or elongation factor 2, the blue molecule, the blue part you can see here. It's not very clear, but please pay attention this part, EF2, elongation factor 2. But in right side, you can see diphtheria toxin or exotoxin A. Like the other toxin we have spoken about them, this is AB type toxin. It is two component toxin. By part B, it is attached to receptor. You can see here by using part B, it is attached to the receptor, and part A is entered into the host cell. Then part A attached to elongation factor 2, and then it is blocked. It doesn't work properly. It doesn't work correctly. Elongation factor 2 doesn't work properly. So you can see here ribosome. The amino acids cannot bind to each other. The amino acids cannot bind to each other. So protein synthesis stops. Protein synthesis stops. So we can say this toxin resulting blocking or stopping protein synthesis in eukaryotic cell through blocking elongation factor 2. Do you understand? If you have any problem, tell me to explain it more. Okay, that's very good. That's very good. Clever student. As I told, there are three types of exotoxins. Exotoxins A, exotoxins S, and C. Exotoxin A is similar to diphtheria toxin, but is less potent in comparison with diphtheria toxin. Exotoxin S and T are extracellular toxins, and in comparison with exotoxin A is less virulent. And one of the other virulence factor of Pseudomonas aeruginosa is type 3 secretion system. And we spoke about it many times. It mediates delivering proteins, effector molecules into target eukaryotic cells, epithelial, which leads to epithelial cell damage, facilitating bacterial spread, tissue invasion, and necrosis. You can see here in this slide better. On top of the slide, this is bacterial cell, and at the bottom you can see host cell. This is host cell, and on top this is bacterial cell, and this is type 3 secretion system. Through this, you can see type 3 secretion system is a kind of secretion system which act as a needle, which act as a channel. Through this channel, through this needle, some proteins, some virulence factors, some effector molecules pass and enter to the host cells, and it can lead to damaging host cell. It can lead to tissue necrosis and spreading bacteria. You can see this is bacteria, this is host cell, type 2 secretion system, and directly through this kind of secretion system, directly these molecules, it can be a virulence factor, effector molecules, some proteins, enter to host cell and damage the host cell. Sorry, what do you mean? Are these fees required for the exam? Fees, what do you mean fees, Shima? So this is type 2 secretion system which help the bacteria to deliver, to transfer virulence factor to host cell and damage their host cell. Now we want to speak about pigment. Pigment, in addition to help us to identify the bacteria better, are virulence factor, or as a component of virulence factor. A blue pigment called pyocyanin, produced by Pseudomonas aeruginosa, catalyzes the production of superoxide and hydrogen peroxide, which are both of them toxic forms of oxygen. This pigment also stimulates interleukin number 8 release, which leads to enhanced attraction of neutrophils. A yellow-green pigment, pyoverdine, it is siderophore that binds iron for use in metabolism. This pigment also regulates secretion of other virulence factors, including ETA, exotoxin A. So these two pigments are, are as component of virulence factor. And about the third pigment, we have no information as virus factor, but maybe in next year's some paper published, and some scientists have good information about their or its effect on pathogenicity of bacteria. Yes, you can see its color here. The first one is pyoverdine, the yellow-green pigment. The second one is blue, and the third one is the reddish to brown. And in some texts or in some articles, Pseudomonas aeruginosa is called a pyocyanic bacilli, as it produce the pyocyanin pigment. No. Drawing, no. I will not ask you to draw this picture. Just this picture, just this diagram help you to understand the content better. You mean, for example, this diagram? No. This diagram, this picture is placed here for better understanding, for better or for illustrating better. And it doesn't need to learn it for exam. Yes. Murtaza, there is no information about its effect on virulence. Just we know the bacteria, Pseudomonas species, produce this pigment, pyrorubin, as a pigment. But we don't know if these pigments affect the virulence of bacteria or no. The scientists don't know anything about its effect on virulence, and perhaps in next year some information obtained. Okay. For Pseudomonas aeruginosa, three pigments are recognized, but it's not necessary that one strain produces simultaneously or at a time three pigments. You know what I say? Let me show you. For example, perhaps one strain of Pseudomonas aeruginosa, just one pigment, or one strain of one or other strain of Pseudomonas aeruginosa produce two pigments, blue and yellow, and the other strain of Pseudomonas aeruginosa produce blue and red, and perhaps one strain just produce red. You know, in the population of Pseudomonas aeruginosa, species, three pigments are recognized, but it's not necessary that all strains of Pseudomonas aeruginosa produced simultaneously or at a time, all of them. Okay, now we want to speak about enzymes. As we told at first, Pseudomonas aeruginosa species, or it's better to say Pseudomonas species, has one of the largest genome, and they produce many enzymes. And these enzymes have important or critical effect on the pathogenicity. Two elastase, LAS-A or serine protease, let me show you in this slide, LAS-A and LAS-B. LAS-A and LAS-B, both of them are elastase. Let me explain it here. Two elastase, LAS-A, serine protease, and LAS-B, zinc metalloprotease. Serine protease, it means it has serine in the active site, and LAS-B, zinc metalloprotease, it means it has zinc in its active site. Act synergically, act together. They help each other to degrade elastin. What is elastin? It's a kind of protein that you can find in some tissue, which, yes, by degrading elastin, resulting in damage to elastin-containing tissue and producing the non-parenchymal damage and hemorrhagic lesion associated with disseminated Pseudomonas aeruginosa infections. These enzymes can also degrade complement components. Of course, you know complement. Complement components are found in blood and are part of immune system and inhibit neutrophil chemotaxis and function, leading to further spread and tissue damaging acute infection. Chronic Pseudomonas infections are characterized by the formation of antibodies to LAS-A and LAS-B, with the deposition of immune complex in the infected tissue. So we have two elastase, LAS-A and LAS-B. Similar to elastase, alkaline protease, the other enzymes, alkaline protease contribute to tissue disruption and spread of Pseudomonas aeruginosa. In all tissue, we have protein. In all our tissue, we have protein. By degrading this protein, we help the bacterial spread. It also interferes with the host immune response. And the last one is phospholipase C. Phospholipase C is a heat-labile hemolyzing. What does it mean? It is sensitive to heat that breaks down lipid and lecithin. Both of them are found in the membrane of the cell, which facilitate tissue destruction, facilitate or help tissue destruction. The exact role of this enzyme in respiratory and urinary tract infections is unclear, although an important association between hemolyzing production and disease has been recognized. So we have three important enzymes: elastase, alkaline protease, phospholipase C. All of them disrupt tissue. All of them help bacteria to spread in the body, to spread in tissue. And the other important virulence factor which help bacteria to be alive, which help bacteria to be survive and make a challenge for scientists. Many scientists, many therapists have challenges to control this bacteria. Why? Because this bacteria is intrinsically resistant. Intrinsically, what does it mean? Intrinsically means basically, naturally resistant to many antibiotics and can acquire, please pay attention to this point, can acquire, they can receive resistance to additional antibiotic through, through horizontal transfer of resistance genes and mutations. Through horizontal transfer of resistant genes and mutation. Of course, all of you know the meaning of mutations, but what does it mean? Horizontal transfer of resistance genes. You can see this slide. Different method of horizontal transfer of resistance genes. And what is the difference between horizontal transfer with vertical transfer? Please focus this slide. As you see here, for example, in part A, we have two bacteria. One of them is donor and the other one is recipient. The donor has a resistance gene to, for example, to an antibiotic, and through conjugation, a recipient bacteria receive or acquire a resistance gene. Or in part B, through transformation, This bacteria acquire resistance gene, in part C through transduction by a bacteriophage. The recipient bacteria receive or acquire resistance gene, and in part B through reduction. So this method helps the bacteria to acquire resistance genes. So Pseudomonas species have the ability to acquire resistance genes by horizontal transfer method. But it's better to speak about the difference between horizontal transfer by vertical transfer here. You can see here vertical transfer in left side. We have a mother cell. By replicating the mother cell into daughter cells, these two daughter cells acquired resistant cells from their mother cell. You can suppose the mother cell has a resistance gene, but by replicating this single cell, into two daughter cells, these new cells acquired a resistance gene. Yes, Hossein Sabah, the lectures or the slides I sent you didn't have these pictures. Last night I modified the slides, so I will send the new slides to you today. Don't worry about it. So this is the difference between vertical transmission and horizontal transmission. Vertical, by replicating a mother cell, replicating a cell which has resistant cell into daughter cells, new cells receive resistant cell. But by horizontal transmission, for example, through conjugation, transduction, the new cell or the recipient cell receive resistance gene. I hope you understand the difference between vertical and horizontal, because in next lessons we will speak about horizontal and vertical, and we will need it. Okay, let me repeat again. Pseudomonas aeruginosa is naturally, intrinsically resistant to many antibiotics and can acquire resistance to additional antibiotic through horizontal transfer of resistance genes and mutations. The main mechanisms responsible for intrinsic resistance. The main mechanisms responsible for intrinsic resistance are found next. In first sentences, I told Pseudomonas aeruginosa is intrinsically resistant. Why? They are intrinsically resistant. Why they are naturally resistant? Because of two reasons. One of them is low rate of movement of antibiotic, low rate of movement of antibiotic, and the other one is rapid efflux of antibiotic. I will show you these two reasons in this picture. I will explain it by this picture, these two reasons. This is Pseudomonas aeruginosa. It's membrane and cell wall. This is outer membrane, and this is inner membrane. In outer membrane, it has some pore or porine. It has some pores or porine, but its pores or porines are so small, so the rate of movement is low. As these pores or porines are small, the rate of movement is low. The rate of movement is low. But reason number two, we have two efflux transporter, single component efflux transporter and multi-component efflux transporter. Single component efflux transporter and multi-component efflux transporter. By using this efflux transporter, bacteria can exit antibiotic out of the yes. Note this lecture, I modified these lectures, and I will send these lectures to you again, the modified version. And of course, I will send next lecture for next session too. So please pay attention here. The size of this porine or pores are small, so the rate of movement is low. And because of these efflux transporter, the bacteria is able to exit out of the antibiotic rapidly. You can see here, exit out of the antibiotic rapidly. So these efflux transporters help the bacteria, protect the bacteria from antibiotic killing effect. And it makes many problems for therapies. In one side, Pseudomonas aeruginosa is resistant to many antibiotics and receive many resistance genes through horizontal transfer and mutation. And in other side, it has low rate of movement of antibiotic and rapid efflux of antibiotic. A third method, a third form of resistance is adaptive resistance. So we have three strategies. Adaptive resistance is induced when Pseudomonas is exposed to environmental stimuli or specific antibiotics. For example, biofilm formation such as in the lungs of a cystic fibrosis patient or on the surface of catheters can trigger bacterial regulatory genes that allow or permit expression of resistance. In the same way, exposure to some beta-lactam antibiotic triggers the expression of AMPC gene in Pseudomonas that results in inactivation of many beta-lactam antibiotics. It is important to recognize that in vitro susceptibility tests can identify resistance caused by intrinsic and acquired mechanisms, but likely would not be able to predict adaptive resistance, underlying the limitation of this lab test. So it is very important to understand, to identify, the isolate the strain which isolate in laboratory is sensitive to which antibiotic. Okay, but we want to speak about epidemiology. Do you have any question? Hey guys, do you have any question, any problems? Okay. What epidemiology? Pseudomonas is an opportunistic pathogen. I have spoken about this term, opportunistic. It presents in a variety of environments. The ability to isolate this organism from moist or wet surfaces may be limited only by the efforts to look for the organism. Pseudomonas has minimal nutritional requirements. It is very important. Tolerate a wide range of temperatures, 4 degrees centigrade to 42 degrees centigrade, and is resistant to many antibiotics and disinfectants. Indeed, the recovery of Pseudomonas from an environmental sources, for example, hospital sink or floor, means very little unless there is epidemiologic evidence that the contaminated site is a reservoir for infection. Furthermore, isolation of Pseudomonas from a hospitalized patient is worrisome, but does not normally justify therapeutic intervention unless there is evidence of disease. And it is estimated that Pseudomonas aeruginosa is responsible for more than 50,000 healthcare-associated infections annually in the United States and approximately 440 deaths. So you can find this bacteria everywhere. And this is opportunistic patient. So patient at high risk for developing infections with Pseudomonas aeruginosa include neutropenic or immunocompromised. Immunocompromised patient with weak immune system, cystic fibrosis, burn patient. All of them are high risk for Pseudomonas aeruginosa. But clinical disease, some diseases are more important. One of them, one of the most important is respiratory or pulmonary infection. And the human respiratory tract is divided in two parts: lower respiratory tract and upper respiratory tract. And Pseudomonas aeruginosa infections of the lower respiratory tract can range in severity from asymptomatic colonization. Asymptomatic, it means no symptom, or benign inflammation of bronchial to severe necrotizing bronchopneumonia. Colonization is seen, pay attention here, just is seen in patients with cystic fibrosis, other chronic lung disease or neutropenia. It means if you are normal, if you have no disease, if you have no problem in your respiratory disease, Pseudomonas aeruginosa doesn't make you disease. It doesn't make you any problem. Infections in patients with cystic fibrosis have been associated with exacerbation of the underlying disease and invasive pulmonary disease. Mucoid strains, yes, just strains which produce exopolysaccharide on its surface, are commonly isolated from this patient and are difficult to eradicate, to remove. Why? Because chronic infections with this bacteria are associated with progressive increase in acquired antibiotic resistance and expression of adaptive resistance. And the mortality rate is as high as 70%. It's very high. The bad news is the mortality rate is high and eradication of the bacteria from respiratory tract is difficult. And the good news is this bacteria doesn't make disease in normal person, and just people who have problem in their respiratory tract um, are high risk. The other important infection is primary skin and soft tissue infections. As I told, these kind of bacteria produce different kind of enzymes which potential um, degrading tissue. So of course they make soft tissue infections. Pseudomonas aeruginosa can cause a variety of primarily skin infections. The most recognized are infections of burn wounds. Perhaps you were involved in burn wounds and you saw some burn wounds which were covered by Pseudomonas aeruginosa, and you smelled a bad smell, and you saw the color of the wound, the green or blue color of the wound. Colonization of a burn wound followed by localized vascular damage, tissue necrosis, and ultimately bacteria is common in patients with severe burns. The moist surface of the burn and inability of neutrophils to penetrate into the wounds predispose patients to such infections. Wound management with topical antibiotic creams has had only limited success in controlling these infections. In some cases, burn wounds, burn is not the main cause of death. But infection due to Pseudomonas aeruginosa is the main cause of death, and it is very important in hospital. The other infection is folliculitis. Folliculitis, another common infection caused by Pseudomonas resulting from immersion in contaminated water in swimming pool. Secondary infections with Pseudomonas also occur in people who have acne or who depilate their legs. Finally, Pseudomonas can cause fingernail infections in people whose hands are frequently exposed to water, who frequent nail salons. And this bacteria is also the most common cause of osteochondritis, inflammation of bone and cartilage of the foot after a penetrating injury associated with stepping on a nail. You can see here a picture of Pseudomonas folliculis. Pseudomonas folliculis. And infection of urinary tract. Is seen in patients with long-term indwelling urinary catheters. Of course, in patients who are hospitalized. Typically, such patients are treated with multiple courses of antibiotic, which tend to select for the more resistant strains of bacteria. We have ear infections related to Pseudomonas aeruginosa, external otitis caused by Pseudomonas aeruginosa with swimming, an important risk factor. It's called swimmer ear. This localized infection can be managed with topical antibiotics and drying agents. Malignant external otitis is a virulent form of disease seen primarily in persons with diabetes or old people. And eye infection. Yes, infection of the eye occur after initial trauma to the cornea, for example, aberration from contact lens, a scratch on the eye surface, and then exposure in contaminated water. Corneal ulcer develop and can progress to rapidly progressive eye-threatening disease unless prompt treatment is initiated. So we should pay attention, and all of them are important. The other infection which are important is bacteria. Yes, bacteria is important. But please pay attention to this point. Bacteria caused by Pseudomonas aeruginosa is clinically indistinguishable from that caused by other gram-negative bacteria. It means you cannot distinguish bacteria which is caused by Pseudomonas from which is caused by, for example, E. coli. Both of them are similar as other, but the main point is here. Mortality rate is affected patients is higher with Pseudomonas aeruginosa. Why? Because we have two reasons here. One, predilection of organism for immunocompromised patients. It means bacteremia due to Pseudomonas aeruginosa is in immunocompromised patients. Two, difficulty in treating antibiotic resistant strains, and three, inherent virulence of Pseudomonas. Bacteremia occurs most often in patients with neutropenia, diabetes, extensive burns, and hematologic malignancy. And most bacteremia originated from infections of lower respiratory tract, urinary tract, and skin and soft tissue. And yes, so we should pay attention to this point. But Pseudomonas endocarditis is uncommon. Or gastroenteritis is uncommon. Or intravenous problem is uncommon. You should pay attention to this point, for example. Yes, for example, I speak about it here. Yes, nervous system, musculoskeletal system. Yes, you can find some people who are involved with Pseudomonas aeruginosa, gastrointestinal tract infection with Pseudomonas aeruginosa, or nervous system problem with Pseudomonas aeruginosa, but they are uncommon. And this book has no many information about it, so I'm not going to speak about it with more detail. Just it points in 2 or 3 lines, and I do point it here. Burn poisoning, you mean toxic toxin? What do you mean burn poisoning? Is it possible for you to explain it more? Will you please help us with next lecture in 1 hour? What do you mean next? We teared and found next lecture. What do you mean you want to? I want to finish this lesson here, and on Tuesday I want to finish this lecture and start new lecture. New lecture will be about Campylobacter, and I will send the slides tonight. But what do you mean, Hussein? You want to find it in 1 hour? 1 hour? What do you want? The third in one. Another session? You want to take part in another session? You are going to take part to another session. You mean here? You mean this? You want to take part to another session? Okay, you have another lecture on 15:30. Okay. Uh, our lesson, our lecture finished now, and you have time to take part for next lecture. Okay, I will send you. And Murtaza, burn poisoning is different from burn wound. Yes, burn poisoning is different from burn wound that I explained about it here. Okay. If you have any question, we can finish the session, and I will see you on Tuesday at this time. Yes. No, I will add